So there was something called the Synthetic Yeast Genome Project. That's an international initiative led by Patrick Kai of the University of Manchester. You can go check out the article in the references in the description of this episode if you want a little more information on that. The point of this so-called Synthetic Yeast Genome Project is to create the first synthetic eukaryotic genome. Now, I'm going to spend some time talking about what that means and why it's significant. But let's just go over some of the specifics of the project itself, and then I'll define some terms in the realm of biology, synthetic biology, just to make sure that all listeners are kind of up to pace here with some of the definitions, some of the words that I'll be using in this episode, in case you don't know them already. Okay, so this is a project. Again, it's called the Synthetic Yeast Genome Project. Why would somebody do this? Why is it interesting to try to synthetically create a genome? Well, it seeks to write a synthetic genome from scratch, and that would enable the design of organisms, supposedly, with novel abilities and greater environmental tolerance. So the point is, we can look at nature, we can see that it comes up with, just an astounding level of sophistication in terms of the things that it creates. So what if we could do that ourselves? You think about things like agriculture, or, even the limbs on our body, or really anything to do with life. Things that we eat, our own bodies that we use to move, our thinking processes, whatever it is, we know that, we kind of have to put up with the solutions that nature has given us. But what if we could go beyond that, right? Whether you're talking, transhumanism, or whether you're talking about, again, something like agriculture. Maybe you can make crops more resistant. Maybe you could make them easier to grow. They could grow in regions where they might not otherwise. Maybe that helps fight poverty and all this kind of stuff, right? And to some extent, obviously, humans have been able to achieve this kind of thing. But the idea to go after a genome and actually synthetically create that, it would be a new kind of control, because the genome is essentially kind of the full blueprint, the whole kind of genetic package of an organism that sets it up to go on and, create the things it creates, to produce the functionalities and the biomatter that comprise its system, allowing it to evolve and go forward. It's kind of the whole starting point, right? So if you could, as a scientist or an engineer, or a kind of mix of both, maybe a bioengineer, if you could come in and actually design the genome of an organism, maybe an organism that doesn't exist yet, or a new genome for an existing organism, if that makes sense, then the idea would give you a lot more control over the outcome, right? Whether those are crops, whether those are biological limbs on a human, whatever. Now, this kind of idea, it really moves beyond traditional genetic engineering, because that would typically involve, kind of the modification of genomes, right? So you would take an existing genome from nature, something that we couldn't come up with ourselves, because, we're talking millions of years of evolution to arrive at something like that. And then we would try to modify it, to kind of tweak it and make it a little more beneficial. Again, maybe it's, like some type of bug tolerant, agriculture, or, something that wouldn't require pesticides, let's say in agriculture, a type of crop that would be more robust, or whatever it is you're trying to make, right? A version of what nature has come up with, but something that we have tweaked to make it a little more suitable for our modern life. Now, in that project I was talking about, Patrick Kai is at the University of Manchester. They've got this, again, they call it the Synthetic Yeast Genome Project. They're doing it, an ideal kind of model organism that you might focus on is something called brewer's yeast. And it's kind of an ideal in this kind of bioengineering realm, because it's got this evolutionary proximity to humans, and it's kind of well understood in biology. So by evolutionary proximity, I essentially just mean, if you look at the evolutionary tree, it seems to be kind of genetically related. Like, it's not that far down the branch or up the branch, however you want to say that, to where humans are. There seems to be a lot of genetic similarity. So if you can figure out something with brewer's yeast, then presumably you could figure out something with humans as well, right? And it seems kind of odd to study something like yeast compared to humans, but if there is some genetic similarity, and you think that going after genetics is a way to get some control over the outcomes in biology, in life, then, it's a lot easier to put a brewer's yeast in a lab than it is a human being, right? So you can kind of find something on the evolutionary tree that is relatively close to you genetically, and then do the genetic testing and design that way. A lot of human cellular processes can be replicated in yeast, so the story goes, and it's just kind of a valuable biotechnology and medical kind of benchmark, if you will, or kind of test subject, maybe is a better word, to use to try to study what you want for humans. Now, the assembling of DNA sequences, and again, I'm going to define some of these terms in a bit, so don't worry if it get a little bit jargony. I'm going to go back, but I just kind of want to put some context around this idea of, trying to create the first synthetic eukaryotic genome. Okay, and I'll define those terms, but I want to just impress upon you why people think this is important, right? Now, assembling DNA sequences is already a known process. When we talk about kind of like synthetic biology or bioengineering, there's already stuff that's been around for some time, and assembling DNA sequences would be, what you'd consider kind of a known process. And that's kind of related to what we're going to talk about. But the genome is a bigger thing. It's the kind of, again, the full blueprint, right? But the primary challenge in designing like a full-blown kind of genomic sequence that results in a viable organism is just a much bigger challenge than just trying to piece some DNA sequences together, if you will. Because you have to understand, well, they believe you have to understand the fundamental design principles of functional genomes. So it's kind of this, hey, we want to do the bottom-up approach. We want to try to piece together this most important part of life called the genome, kind of the full blueprint genetic package. We want to do that from the ground up. Because if we could, then we would know what all the pieces are. We would know how all those pieces interact. And we would truly have control over the outcome, so the story goes. So if you just think about assembling DNA sequences, right, that's fundamental and normal to synthetic biology and genomics. It's achieved when you use a combination of chemical synthesis, molecular biology techniques, and, a kind of a range of bioinformatics tools. We don't need to get into all that and what that means, but just understand that that's kind of standard, right? But this group at this international initiative, the Synthetic Yeast Genome Project, coming out of University of Manchester, they want to understand the full design rules, right? They want to go beyond just kind of piecing little pieces of DNA together, or maybe having some kind of control over chromosomes, and go all the way to the genome, that full kind of package of genetic material, right? They want to understand those design rules, how genes and non-coding DNA kind of interact to create functional, viable organisms, okay? And the way that this initiative is undergoing this, and again, you can kind of go to some of the original papers, and you can go to the original article that kind of mentioned this. Again, I'll put those references if you want to know more, but they basically explore the vast, what you might call combinatorial space of the yeast genome efficiently. And they do that by employing a tool called Scramble, and that allows for the randomization of yeast genomes on command, which would facilitate the study of genetic components and their function. So in other words, you want to go after the design rules. You want to know how to piece together a genome, but there's so much unknown about that, right? Nature is exceedingly complex. It's hard to know how all these pieces come together. So one way to try to go at that is to introduce a good level of trial and error, and that would allow you to mix and match a bunch of different things, and then you kind of, after the fact, try to validate that what you have is viable, right? So you mixed and match, and 99% of your mixing and matching experiments didn't work. 1% of them seems to, because it seems to have produced a viable organism or part of an organism, or however they are going to experimentally kind of validate that the genome is doing or is what they want it to be. Okay, so that's that. Now let's get into some terms here, because if you don't have much of a background in biology, or it's been a while since you've done it, most of you know DNA, but chromosomes and genome, what's the difference? Yeah, eukaryotic cells versus prokaryotic. Let's just do a bit of back to the basics on biology. Let's get these terms defined, and then we'll bring it back to the kind of larger context and the more broader perspective science conversation that I want to have, and my opinions and commentary on this particular project. Okay, let's start with DNA. So we've all heard of DNA. I mean, it's in every crime podcast and movie and show, and that word, and you probably learned about it in high school and all this kind of stuff, right? So DNA is deoxyribonucleic acid. That is a long, double-stranded molecule that carries the genetic instructions used for the growth, the development, the functioning, and the reproduction of all known living organisms, and even many viruses. So it's kind of like the fundamental informational thing in biology that gets used to kind of set up the processes going forward, to build the proteins, to build the muscle tissues, to build all the things that have functionality, really to build everything we associate with life, right? And then even some viruses. So that is the fundamental strand of encoding of information, and all life kind of follows the rules in such a way that as long as you have these, essentially the kind of nucleotide pattern, such that it is, then the rules of life will kind of programmatically set up a situation to produce what's needed. Okay. So yeah, it's almost like having something on a disk drive, right? It's kind of like the biological counterpart to, having data stored on the hard drive, which would tell you how to set up a process. And once that process kicks off, it would have all the information it needs to essentially build what it needs to build. Okay. So that's DNA, and we all have DNA in us and other living organisms, and you can identify things through DNA, and it's kind of like a fingerprint of the organism that you take it from. Okay, so that's DNA. I think most of us know that. The next one I'll talk about is genes. So what's the difference between DNA and a gene? Well, a gene is just a specific segment of DNA that contains the instructions for making a functional product, such as a protein. Okay, so the DNA is this, again, a really long double-stranded molecule, but you don't need to use all the DNA to build all the things at any one time anyways. Though if I'm going to work on muscle tissue, if biology is going to work on muscle tissue, it doesn't necessarily need all the DNA to do that. It's going to use certain segments to do certain aspects of muscle tissue. If it's making neurons for the brain, if it's making fingernails, right, parts of the eye, there are going to be specific segments within the DNA that are more responsible for kind of coding and ultimately producing that type of biomatter. So the full DNA is the one long big molecule that has all the information or instructions necessary, and there are specific segments within that DNA that go on to code or produce certain parts, let's say, of the body or whatever organism we're talking about, right? That's the gene. The gene is segments of the DNA. And then I said the genome, right, which is what we're getting into, and I kind of alluded to this earlier, that this is kind of like the full blueprint, the big thing. And so the genome is the entire collection of DNA in an organism. Now that can consist of only a single DNA strand in some cases, like prokaryotes, or many DNA strands organized into chromosomes, which I'll define in a second. And that would be the case of eukaryotes. So humans, we have 46 chromosomes, so their genome is made up of 46 distinct DNA strands. You could compare this to diploid cells and haploid cells. The human genome contains approximately 20 to 25,000 genes. But anyway, okay, so what is the genome? The genome is just the entire collection of DNA in an organism. Okay, regardless of how that DNA is packaged up, and forget all that for a second, just if you were to collect it all in that organism, the complete kind of genetic material of that organism, it would be the genome. Okay. And then we have the chromosome. I said, okay, so what is the chromosome? Well, a chromosome is a compacted structure of both a single strand of DNA along with other things like associated proteins, and that allows the DNA to fit inside the nucleus and function effectively. So essentially, a strand of DNA by itself is not really that useful, even though it's got all the information that you need to go on and produce things, because it has to be stable, it has to be protected, it has to find its way into the proper part of the cell. It needs a vehicle to do those things. It needs to have its functionality exposed correctly. So the vehicle that allows DNA to do what it does would be the chromosome. It's like putting a jacket around it so that it has an interface to it, for lack of a better way of putting it, so that it can do what it's supposed to do. Okay, so it's just, you take the DNA and you put a bunch of proteins around it, and now you got a chromosome, which enables DNA to fit inside the nucleus and function properly. Okay. So they're like vehicles for DNA. They ensure that the genetic material is protected, it's organized, it's efficiently transported or accessed within the cell. Humans have 23 pairs of chromosomes, 46 total. 22 would be the non-sex chromosomes, and one pair for the sex chromosomes, right? XX for females and XY for males. Each chromosome contains a single linear strand of DNA, and then all that other protein stuff, right? And because genes would be a segment of the DNA, then that strand would include genes, and you've got like regulatory regions and non-coding sequences within there and all that stuff. Okay, I'm going to do a quick recap of all this just to put this all into perspective. But again, I just want to kind of define some of these terms. We hear terms like prokaryote, right? They're doing eukaryotes in that study. Prokaryote, what's the difference? A prokaryote are just cells that do not have a nucleus or well-defined nucleus, right? You kind of have genetic material just organized differently within the cell. A singular circular chromosome as opposed to what we would see in the eukaryotes. The eukaryote, the cell, has a well-defined nucleus where the genetic material, the DNA, is stored and protected. You got things like viruses that they're not prokaryotic. In fact, viruses are not really classified as cells at all. They don't really fall into the categories of prokaryotes and eukaryotes. You've got bacteria, which would be prokaryotic. They are single-celled organisms without a nucleus or membrane-bound organelles and all that kind of stuff. So if you hear these terms like viruses, bacteria, genes, DNA, genomes, eukaryote, prokaryote. So I just want to kind of define those terms. So let me just give you a quick summary, and then we'll get back to the story, okay? So just kind of the back to the basics biology, make sure we're all on the same page here. So the most basic unit of information in life is the DNA molecule, okay? It's a long, double-stranded molecule that carries the genetic instructions used for the growth, development, and functioning, ultimately, of the organism, right? Now, DNA alone would not really be that useful, so it has to get packaged up in a chromosome. And that's a compact structure, which has the single strand of DNA that we just talked about, along with other proteins and things that are needed to kind of package the whole thing up and make it functional. It allows the DNA to fit inside the nucleus and function effectively. Now that the DNA can function, thanks to the protection and utility of the chromosome, specific regions in the DNA can be used to code for things like proteins. That's because to create new molecules, you don't need the entire set of DNA or the entire strand. These specific regions are called genes, and those are specific segments of DNA that contain the instructions for making a functional product like a protein, right? So if we take the entire unique set of genetic material in a living organism, we call it a genome. A genome is the entire collection of DNA in an organism, kind of like its blueprint. So that includes all the genes and non-coding regions. In humans, we have 46 distinct DNA strands, right? Now, they're identical strands, actually. They're just repeated 46 times. So they're kind of like spatially distinct, not molecularly distinct, if that makes sense. And those get organized into the chromosomes. So if you want to, so here's the point, right? If you want to create an organism from scratch, you would need to create the genome, since it is this level of abstraction that is believed to contain all the information necessary to start, grow, and just get this thing to do what you need to do, right? Start, grow, and evolve the organism, right? Now, a kind of simple way to just think about all this is just in terms of quote-unquote size. You could say that the order of size would be the gene would be the smallest because that was a segment of the DNA strand. The next would be the DNA, which would be the full kind of fundamental strand, right? Then it would be the chromosome, right? Putting the jacket around it so it can do what it's supposed to do. Then it would be the genome, which would be kind of the full collection of all the chromosomes and everything that it contains. And then ultimately the organism, right? Of course, between genome and organism, you could put like body parts or organs or things like that. But in terms of the words that we're talking about here, think about it as gene, and then DNA, and then chromosome, and then genome, and then ultimately the organism. So you can see that, essentially right below the organism is the genome. So that's why this project, if you really want to get control over the biomatter, right, over, maybe a living organism or some aspect of life, some aspect of biology, then I guess the kind of the holy grail there would be able to design the genome, right? That would be the point. And just a little more kind of summary of this stuff to think about it as a process. Think of it as, DNA is driving the protein synthesis, right? And as per something called the central dogma of molecular biology, it's kind of like the computational pipeline that happens. You could think of it as. So DNA stores genetic information that gets accessed and executed through a computational pipeline where first you have one, transcription. That's where the DNA is read and transcribed into messenger RNA. Then you have translation, step 2, where the mRNA gets translated by ribosomes into a sequence of amino acids that forms the protein. Number 3, you would have essentially folding and functioning, right? So proteins fold into specific three-dimensional shapes, and that enables their biochemical functionality. So depending on the shape of that protein, that's what it ends up doing. In other words, if I change the three-dimensional shape of a protein, it's going to end up, doing or making something else, right? So proteins synthesized based on DNA instructions from the building blocks of new proteins that are synthesized in this process, right? Based on those DNA instructions, they form the building blocks of new biomatter. So when we're talking about tissues and enzymes and, signaling molecules, whatever it is, that's where that comes from. Okay, so that was our kind of back to basics bio 101. Hopefully that helped you understand, if you didn't already know, the difference between the DNA and the gene and the genome and the chromosome and the eukaryotic versus prokaryotic cells and all that good stuff. Okay, so now when we go back to, why would people care about trying to create the first synthetic eukaryotic genome? Hopefully you kind of get that now, right? If you could not just rely on biology to come up with the blueprints here, if you could create that genetic material from scratch, the argument is you would have a lot more control over the things that, we do, the things that we eat, the limbs that we have on our body, some type of life outcome, right? You could use science to help control that better. Okay. So Let's talk about this now. So, what's my kind of commentary on this? What's my opinion about this? What do I think about the science? Well, one thing you can say for sure is that you should expect major unforeseen consequences in an experiment or an effort like this. So this notion, on its face, it seems to make sense to a lot of people. If I say, this is what a genome is, and this is how it sets up, all the proteins and all the things that get made, in the human body or in, the crop or whatever it is. And then I say, maybe we can actually design the genome ourselves. A lot of people would think, oh, that sounds like you have a lot of control over things, right? That sounds like you would really be able to tailor the crops or someone's organ or someone's limb or, whatever. You would be able to tailor that to suit what we need in life. It sounds like a good thing to do on its face. Why wouldn't you use the knowledge of science and engineering to be able to do these things? The problem is really in this notion of design and this idea that we have a lot more control over these things than we actually do. So designing an entire genome that functions cohesively to produce a living, viable organism or even something en route to that, that is a far more complex challenge than, say, just synthesizing or editing part of DNA. Okay. Now, they're saying it requires a deep understanding of how every part of the genome works together to drive life processes. Well, you could say that's the challenge, which in and of itself might prove impossible, but it's also problematic to just think that that is the challenge. Just because you know how all the pieces, what all the pieces are of the thing you're creating and how those pieces interact doesn't mean you can actually make the thing. Now, that sounds odd to a lot of people because when we think of, like an automobile or an internal combustion engine or a train, or really most of the things we have engineered as humans through human history, it was exactly that, right? Everything has pieces and those pieces bump into each other in a causal fashion. And so if you follow that chain of events, you get the outcome. So if you can reverse engineer something and see the pieces and you know what every role plays and you can reproduce those roles and blah, 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 then you can just make the thing. But that's not how biology works. That's not how life works. And we know this because if you look at genuine complex systems, genuine complexity, they're very, very different than that. They operate via emergence. They don't have this deterministic chain of events that produces their outputs. So even if you reverse engineer a complex system, and even if you could feasibly, which is already kind of doubtful, but even if you could identify all the pieces and you think you could define the roles of those pieces, and even if you think you could connect them up the same way, that still doesn't mean you're going to get the same result. There are just way too many interactions, and those interactions are not necessarily deterministic and causal the way they are in something like a steam engine or something that we built during the Industrial Revolution. Most of the machines and, let's say rules-based software that we create. So the whole premise is quite flawed. The idea that you can design the genome, the idea that you can identify the pieces, put it together like Lego blocks, and then get the end result. That's just not how life works. That's not how biology works. That's not how complex systems function. They don't function as a sum of their parts. Okay, so even if you have the parts and you create the sum, that's not going to give you the output the way the genome actually functions, right? And it sounds a little bit mysterious if you're not familiar with emergence or studying of complex systems. That might almost sound a bit hand-wavy, like, well, what do you mean? What is it? Well, that is kind of the great mystery, right? The point is it's not a deterministic thing. The properties that exist at the larger scale are not found in the pieces from which that system is comprised. Okay? The whole is much greater than the sum of its parts. A new thing happens at different scales, right? So this notion that you're going to be able to design something like a genome is highly problematic because it's not subject to design. It's not something that is arrived at by piecing pieces together and getting the things to interact with each other in a deterministic fashion to produce the outcome. So that's already a bit problematic, is that word design, right? The whole premise of a project like this really just assumes that, whatever operates at the higher level is doing so in a deterministic collection of the pieces it's made from, and that's just incorrect. That's just not true. You'll never find an example of a complex system that is like that. If you find a system that's like that, it's not complex. But life is complex. We know this, right? There's no greater example of genuine complexity than that, than what we see in life. So we really, we see emergence at all levels, and really at all levels. I mean, even a molecule is not a deterministic system, right? I mean, you can't even go beyond, the so-called three-body problem without running into the issue of things not really being deterministic. Now, you can kind of get away with it if the system is simpler. So if the molecule is a lot smaller, it still has, levels of emergence and genuine complexity, but because there's only so many pieces, you can kind of approximate more effectively, right? This is why things like, quantum or computational chemistry or computational biology, when they start to try to do, let's say, electronic structure calculations on molecules, if the molecule is not too big, then the approximations are pretty feasible. As soon as you get into any reasonably sized molecule, the approximations become extreme, so extreme that sometimes the calculations become kind of nonsensical. And that's just because now you're genuinely entering the realm of emergence. It's just not a sum of its parts. It's not a collection of its pieces. Now, that's not to say there aren't modeling approaches and mathematical techniques to try to operate at the emergent level, but it's important. But again, going back to what we're talking about in this episode, this notion that you can design something piece by piece in biology is pretty borderline nonsense. It's just not how complex systems work, right? Now, in fairness to the researchers, right, let's say they might still appreciate that. And maybe, if you think about the fact that they're using the scramble tool to mix and match things, at least they are embracing trial and error, right? They are realizing that, we need to take kind of this trial and error approach to do the mixing and matching rather than meticulously piece together all the pieces because we think they know how they're supposed to interact. So that is better. That is promising. That is at least recognizing that the space of possibilities that you have to explore is so vast that you can't just use a bunch of upfront knowledge to design the pieces of a genome, right? But there is still an issue here because rather than kind of operating in the possibility space, which would be all the possibilities that you can mix and match, they're actually operating in a design space. And that means they're choosing the limited set of pieces that get mixed and matched, and they're choosing how they get mixed and matched. And so you're still operating this kind of very myopic and narrow space of possibilities, which is conforming to your designs, the design of your experiment, and the knowledge that you think you have about how things inside the genome interact and operate. Not that all that knowledge is wrong, but when you frame it in terms of design, it becomes really problematic. You think you know how pieces come together to produce the output, but it's not even the right premise. That's just not how biology works. Things don't use pieces and that interact in a deterministic way to produce outcomes. It's just not the right premise, I would argue. So here's what actually the researchers should expect. You will have unexpected emergent behaviors, right? So you might get metabolic bottlenecks, feedback loops, and chaotic dynamics that just never could have been predicted in the way that they rise. So now think about that. So you're piecing something together with whatever knowledge you have. You're trying to quote unquote design the outcome, even though you're embracing a tool like Scramble. And you're just going to get all kinds of unexpected things, extreme levels of unexpected behavior, which obviously flies in the face of design itself, right? The whole point of design is to use your knowledge to produce some known outcome, and you're getting a ton of known outcomes that are going to come at unknown times and in unknown ways, right? You could have a design gene that may work perfectly in isolation, like in the lab or close to perfectly, but it could disrupt the system's balance when introduced into a functioning genome, right? So you do some isolated thing, and that's my one piece that's going to go into my design genome, and it works as expected, and then you go to put it in, you think it's good to go, and then it doesn't behave as expected, right? It's somewhat analogous to building software where you can try to create this perfectly functioning component, which in isolation does exactly what it's supposed to do. And then you go to place that, let's say it's like a REST API or some component into the bigger embedded system of software. And then things just start to go haywire and they don't go that well. And that's in deterministic software, which is not even complex, right? That's just complicated. That's not complex, right? Complicated is not complex. In genuine complexity, you have no idea what those interactions are going to be. You have no, the feedback loops, the nonlinearities, all the emergent properties that manifest in complex systems. So it's a completely different story altogether, right? The level of unexpected change. Even small changes we know can cascade into large unpredictable effects due to feedback loops and nonlinear interactions. Like I said, you could let's say, change regulatory elements for one gene, but that might unintentionally affect dozens of others, which could ultimately destabilize the system. A synthetic genome designed in one environmental context could actually completely fail in another. And there's no way you could test all environments, right? Or even replicate the environment that you think it's going to be in. It would be very hard because the genuine environment that it gets embedded in is just so vastly complex and has just so many unknowns and tons of nonlinear interactions and all this kind of stuff. The project cannot feasibly test every possible condition for viability, right? So what does viability even mean? Natural genomes, the ones created by nature, they often include redundant and degenerate systems, right? You could have like multiple genes or pathways that perform overlapping functions just to ensure its robustness. But think about how different that is in engineering, right? Synthetic designs would actually aim for things like minimalism and efficiency, because that's what we do in engineering. It would strip away the redundancies because they think that they're not needed. It's kind of like the whole nonsense around junk DNA, which for years people thought, let's say the non-coding regions or parts of DNA that just didn't seem to do anything, that maybe they're just kind of like a vestigial thing from evolution and that you don't need them. Well, that's stupid. That never should have been assumed, right? With a proper understanding of how, the variation, iteration, and selection works in evolution, nature does not hang on to things for random reasons. Those are definitely there for a reason, even if you don't know what that reason is. No one should have ever called parts of DNA junk DNA. There was never a reason to have that as a theory. Anyways, that gets into just the problem with the current scientific paradigm and how people get that backwards, but we won't get into that now. But synthetic designs are going to aim for things that engineers aim for, right? Minimalism and efficiency, like rules-based software, right? Like what an engineer would normally do. Redundancy is seen as bad. Complexity is seen as bad. That can make a system, but if you strip that away, that can really make a system fragile and prone to failure. When that system ends up becoming confronted with environmental stresses, or things like mutations and things like that, it's not an engineering problem. Bioengineering should not be taking things from Industrial Revolution engineering, right? It's just a completely different realm that you're in, but inevitably that happens. So that's what researchers should expect. And I think that there's a naivete to this type of project. Not here to just pick on this project, right, from this university. You see this kind of thing all the time, right? It treats it like a technical engineering problem, and it's just not. That's not to say that there aren't things that can be modeled, and that there's, the science can't push the needle on these things, sure. And I'll talk about that in a bit, but if you're not going to design a genome, right? You might say you did it, and then you do a genome, and you came up with something, and then you say you designed it, but there's no reason not to expect that it's going to be catastrophic. That's not going to be catastrophic. It's going to have all these absolute unforeseen consequences because that's how complex systems function. So just talking about the scientific paradigm in general here, look, you cannot piece together the whole under complexity, pieced together. Or if you do, really, you just can't. You might think you did, but when you have that many unforeseen consequences, then that's not really what you did, right? If I tell you that I'm going to design something, and so I use my knowledge of interactions and pieces, and I piece it together, and I go, look, I do it, and then a year later, this thing explodes and causes a ton of unforeseen consequences, whatever that means, right? Or it causes disease, or it causes whatever. It could be a million, a billion, a trillion different things. We just don't know. And they're not even necessarily all harmful, but let's just say there's all these other things that happen. Well, that's not me designing, right? You can't really say you designed. You really just kind of mucked about, kind of got the outcome you wanted, and then there was just an absolute ton of side effects and byproducts, right? I mean, really, even the term side effects is pretty misleading, right? It's like in medicine, like does medicine have side effects? Well, it just has effects. And one of those effects we choose as beneficial because we like what it did, right? If I take the Advil and my headache goes away, that's still a side effect, right? It's still just an effect. I just chose that one because that's good, but that doesn't mean there's a bunch of other stuff not going on, right? So the notion of design is really problematic when we talk about, especially in things like biology, right? You just can't actually piece together the whole, even if that's the narrative you're applying to it. So our current scientific paradigm is just steeped in this reductionism, right? Where most areas of science are running off the assumption that you can reverse engineer a system, inspect its parts, and then get an idea of how the whole works, which is just nonsense. It's just not how it works. Now, and again, I said like their tools scramble. On one hand, it's kind of promising because at least they're admitting they've got to embrace the trial and error. Okay, I like that part. It's not like they're actually taking nano tweezers or something and piecing together, right, like individual molecules or segments of DNA. Or they might be doing that too, but they are embracing some, kind of heuristic, high-level approach to try to mix and match. It allows them to, but what they're probing there is the design space of genomes, which is a human thing. It's not a life thing. A human meaning, I mean, humans are life, but I mean, it's a human concocted thing, right? Where we create the design space. It's not the true possibility space that biology is operating in, which is vastly much larger than that. So they're going to explore those configurations and try to land on desirable traits for, supposedly improving kind of organismal function, right? But that mixing and matching, again, alone is a great approach, but they can only ever mix and match their designs as it's exceedingly limited compared to the full possibility space that you would find in actual biology, actual life, right? So it just bakes in the assumptions about inserting components and the roles that those components supposedly play. So anyways, very reductionist, very causal, very deterministic, very counter to complex systems and how life functions. So what's the broader perspective here? Let's just step back from this and let's just do a quick recap. Okay, I did, just to put this all into perspective, I said, and this is not to pick on this project. This is just one project, right? There's lots of things like this. This one's called the Synthetic Yeast Genome Project. It's based out of the University of Manchester. And it's trying to create the first synthetic eukaryotic genome. Again, why would you want to do that? Well, because if you could do this from the ground up, presumably you'd have a lot more control over the way things genetically happen in life. And if you do that, you could work on crops, you could work on people, you could work on medicines, you could work on all that kind of stuff that has to do with life. So you can see why someone would want to do that. And then I said, we just kind of quickly define some terms there, right? The DNA, the gene, the chromosome, the genome, right? Genome kind of being the higher level of complexity there. So if you can get that, now you've got the full package and how that goes into cells and all that kind of stuff. And then part of that, kind of computational pipeline of building things. And then I said, okay, but what's actually happening here? Well, it's a bit ridiculous to think of this as design because absolutely, without a doubt, there will be a host of unforeseen consequences. And we know that because that is a universally true property of complex systems. You don't have the kind of control you're pretending to have. I'm not saying you don't have any, but you don't have anywhere near the kind of control that would justify the word design being used. All kinds of small changes can cascade into unpredictable effects. There's no way you're going to be able to replicate the environmental stresses that things are going to be embedded in, whatever it is you're making, in this case, the genome. All kinds of assumptions about, what operates at the higher level is doing so as a deterministic collection of the pieces that it's made from, which is just not true. What happens in one environmental context could easily fail in another. You just can't test for those conditions and viabilities. Feedback loops, bottlenecks, chaotic dynamics. And this is not to kind of poo-poo research or science directed in this general direction. In general, it's just going after this notion that you can do it from the bottom up and you can design. I promise you can't. But it's unsurprising that this kind of finds its way into the scientific paradigm. Okay. So again, what's the broader perspective here? It's too much kind of physics envy, right? This is a term usually you apply it to kind of the social sciences where maybe someone in economics is kind of acting as though the economy can be modeled and predicted with basic physics, right? Which is just not true. Because again, the economy is a complex system and it just, it's not reductionist. It's not deterministic like that. It's just not how things work. But it's convenient to use the simple kind of physics math, not that all physics math is simple, but the simple version of the physics math to try to frame what you're doing. You can do this socially. Right? It's like social engineering. It's like, hey, we can make society better if we fit it to the rational rules of mathematics or something, right? Or what we know about, the way machines are supposed to operate. And of course, that leads to absolute catastrophic consequences because that's just not how it works. And there's all kinds of historical precedent for this. So that's physics envy. You could even call it engineering envy because the overwhelming majority of engineering is this kind of very simple rules-based stuff. And that's just not how life functions. And then, of course, there's even ethical challenges there, too, especially when you have the historical precedent that tells you that framing social issues or any complex issue like this, including biology, is just really, really problematic. It has a lot of side effects, so to speak. So it highlights the need for understanding and engaging with complex systems across disciplines. Okay, engaging with complex systems across disciplines. And you would assume that biology wouldn't maybe be making this mistake, but they do. They have the physics envy as well, because people tend to think that if you do things the way physicists do it, which is to use a lot of math and to use kind of very quite frankly, simplified versions of life, that it tends to sound more rigorous because you're a lot more precise and you've got all these equations. And when you step outside physics, that just starts to fall apart. That's why you don't see as much math and physics in things like chemistry, and even less in biology, and even way less in the social sciences. It's not because those people don't like math, it's because the phenomena don't fit the math. Not that math can't be used at all, but the way that it gets used in most of physics in that very deterministic, very precise fashion is absolutely not suitable to phenomena that exist at higher scales. The principles of complexity theory, you got nonlinearity, feedback loops, emergence. Those are increasingly relevant in the designing and managing of large-scale systems. Although, again, I don't like that word design. It's not really designing the systems at all, right? It's a completely different thing. I'll allude to that. I'll talk about that in a second. But look, not to mention that, from an ethical standpoint, right, designing synthetic life really raises profound ethical questions about humanity's role in nature and, the potential risks of unintended consequences. And that's not like just a moral ethical thing. I mean, even if you're talking about ethics, you're still stepping back and saying, look, we're just looking at how complex systems operate. This is known stuff. These are general properties. We don't see violations to these properties. And yet you're charging ahead as though you can design this stuff, right? Those unexpected consequences should be absolutely expected. So what can we do about this? How can we kind of think about, what would be a better technique? What would be a better approach going forward in a space like this, right? Well, if you want to actually understand something, right, understand its existing behavior in environments rather than defining quote unquote understanding in terms of how the pieces supposedly interact. Okay, very much today's scientific paradigm is all about kind of reverse engineering, right? We think it's better to peel back the layers and define things precisely, but there's a real cost to that precision, and that cost absolutely explodes when you enter the genuine realm, the realm of genuine complexity, right? So you have to redefine what you mean by knowledge and understanding in that space. If you're a researcher and your area of science, think about your area of science and consider the behavior of the thing, not its components. It's a different way of thinking about something. Think of the behavior of it. So you're going to step back, think of it from a higher level, and how does this thing behave in its environment, as opposed to trying to peel back the layers and try to inspect the pieces, right? That would be a better version of understanding, actually, a better version of knowledge. Consider not making any changes to the organism other than its environment. This would be a way to think about it. Then once, let's say in this case, the organism adapts to the new environment, you could look at the genetic changes that happen naturally and study and understand those. Right. Now, this is not entirely new. This is kind of, you might call this adaptive laboratory evolution, ALE, or even just kind of evolutionary engineering. The idea is that you would create a controlled environment with specific selection pressures, right? So you'd say, well, here's the temperature. Maybe we're going to limit the nutrients. We might even expose the environment to some toxins or expose the thing in the environment to toxins. We could alter the pH levels, right? And then over multiple generations, you would get these natural mutations and recombinations that occur within the organism's population. If an organism's, evolution takes too long, then maybe you do this through computer simulation or even deep learning, right? So it's not, you can use flies that have a lifespan of 2 weeks, and then you can kind of more feasibly study the lifespan, but or the evolution, because they're generationally passing on things on every 2 weeks. But a lot of times that's just not feasible, right? Obviously, with humans, we're going to live 100 years. Or 70, 80 years on average. So that's going to just not be feasible. And a lot of life is like that. But that's where you get into computer simulation. That's where you get into approximations, or, now with AI, right? Deep learning, it can do a lot. So you try to get that kind of generational evolutionary knowledge computationally. But the point is that you're not trying to design or piece something together piece by piece. I said earlier, I'm not trying to poo-poo any level of science in this direction. I'm just saying the design is the bad way to do it. It would make more sense to observe the behavior of a thing in the environment that you think you want it to be in, and then see what evolves over time. Okay. Not to say that there aren't still going to be some consequences to that there isn't some still, a level of naivete, because, again, it's hard to replicate the environment. And are you really replicating the environment? There's still issues there. But it's a much more natural thing. You're still accepting the complexity of life and the way that it works. You're not trying to design it. You're allowing it to evolve. You're just putting some control over the environment and seeing what evolves after the fact. And just in terms of everyday decision making, just accepting what nature has already given us, which is always going to be far more sophisticated, and choosing the environment will allow us or things to adapt more naturally, right? I mean, just in everyday life, imagine, obviously, if you want to be a fitter individual, then you could change your environment such that you would be essentially forced, for lack of a better word, to adapt that environment. Whether that means you're going to the gym all the time, you're hanging out with people that are physically active, right? You will be a byproduct of your environment, right? Show me your friends, show me your future, kind of thing. So this could be work environments, health environments, whatever it is. You're allowing you're not getting in the way of nature. You're not pretending like you can piece things together. You're just saying, okay, this is how nature works. This is, we have to accept the way complexity happens, but it is an adaptive process. Maybe we can do something with that adaptation to get something to evolve in a fashion that is maybe more reasonable. Not that there's no risks there, not that there still isn't naivete there, but it's a lot better than, at least that's respecting the way things actually happen in life. Whereas designing something from the ground up is just simply not how it happens. Okay, I think that's it for this episode. So, I hope you enjoyed that. I hope you learned something. If you have any questions, reach out. If you want to support my show, Science in Perspective, you can quickly and easily support me directly by clicking on the support link that you see in any show description. $3, $5 a month, whatever it is, it goes a long way to help make these types of shows feasible. If you want to be a more dedicated listener, you can actually become a member over at scienceinperspective.com. So in addition to having a one-stop shop for all the episodes, you can find premium content, a member chat room, periodic Q&A sessions with yours truly, that kind of stuff. More specifically, the premium content gets me going deeper into the patterns and the stuff that I talk about in these episodes. Okay. So, in this case, we're talking about things happening with, attempting to design life. And I was giving you kind of a better approach going forward, for the scientific paradigm, putting things in environment, letting things happen naturally. And then after the fact, looking at what has evolved. We're going to go deeper into that kind of pattern and what that means. You can learn a little bit more about that type of science, that type of mechanism, and even just think how you could apply that type of thing, just to life in general, right? To be a more rational, intelligent, acting, and logical person, I would argue. So if you'd like to become a member, head on over to scienceinperspective.com and become a member today. Okay, that's it for this episode. Thanks so much for listening. Until the next one, take care.